Hi, everyone. We're just going to get started. I know people are still trickling in, but in the interest of time. So welcome to the very special session. This is the Basic Sciences session that myself, Christina Guzzo, I'm from the University of Toronto Scarborough, and my co-chair, Jimmy Dikeakos from Western, have put together. We hope you'll see we've got an outstanding lineup of speakers here who are all modeling HIV in vivo in very different ways. And so what we're going to do today, we'll give them each the opportunity to share some of their work and their model systems. So we'll go through each of their talks without a question period. So just get through the 4 talks, and then we'll open it up for one general question period with everyone in the room at the end. So I'll turn it over to Jimmy.
Thank you. So our first speaker is Dr. Kerry Lavender from the University of Saskatchewan. Kerry?
All right, well, thanks very much for the opportunity to talk about our model. I'm trying to go back here. There we go. So I don't really have a lot of conflicts of interest to talk about. I guess I should mention I do work with OncoImmunin down in the US, but I'm actually not going to talk about that data today. What I am going to talk about are our humanized mouse models for HIV disease in particular. We do a number of other models as well. And how we've been using them to study HIV, and particularly to try to develop some new strategies, new therapeutics, and also strategies for cure. So the primary model that we use in our lab for the HIV work right now is the TKO-BLT model. The name seems a little archaic if you're not in the field, but they're usually pretty easy to understand. The initial acronym is usually the strain of the mouse that you're using. So in our case, it's a triple knockout mouse, we call it. And what this is a black 6 strain mouse, and it has 3 different genetic knockouts, hence triple knockout. And these different mutations make that mouse highly immunocompromised, and that allows us then to put a human immune system back into the mouse where we can infect it with HIV and study it from there. So how do we put that human immune system back in? We use what is called the BLT method. So that's where that name comes from. And BLT stands for bone marrow. So we give technically a bone marrow transplant. And then liver and thymus, which are the 2 tissues that we use. So we get our hematopoietic progenitor cells from the liver, and we also implant a little bit of thymus under the kidney capsule of that mouse. And when we put all of that together, then what we get is a mouse that has hematopoietic progenitors in the bone marrow that are going to then undergo hematopoiesis. We're going to get all the different immune cells that you would find in a human immune system coming out of the bone marrow. And that includes some immature T cells or thymocytes. And these then, because we have implanted that little bit of thymus from the human as well, that grows up on the kidney capsule to produce a surrogate human thymus. And these immature thymocytes then can migrate to that thymus, and they can be educated on human MHC so that they're quite functional. And so this is a little bit of a unique thing to the BLT model. The only other humanized models that have that kind of education are ones where they've transgenically expressed human MHC into the mouse. So this results in a highly complete and functional human immune system in the mouse. And we've previously shown that it supports HIV infection and shows multiple hallmarks of the disease. What do I point this at to make it go forward? Anywhere? Oh, there we go. So I don't know if I went 2 slides or not there. This is not good. Oh, we didn't. So the TKO model is one of many humanized mouse models. There are other BLT models, but there are a couple advantages to our particular model, and it's based on that TKO mouse strain. And so one of the advantages is that we retain complement in our model. So if that is important for the type of study you're doing, and it is important for studies we plan to do in the future, our mouse model allows you to do those studies. So there are other very good BLT models out there using mouse strains based on the NOD-SCID background, and all of these have a C5 complement deficiency, and so consequently, if you want to have complement fixation in your model, the TKO may be the way to go. Another advantage particular to our model is that we lack or at least we have substantially limited graft versus host disease, or GVHD. So what is GVHD? Well, the problem with having a very functional human immune system inside of a mouse is that immune system ultimately starts to attack the mouse itself. And so this is a fairly significant issue, primarily in the BLT models, because they're so highly humanized. And so you can see here, so this is a nod-skid gamma mouse, so that nod-skid background. Can be quite bad in these mice. So they look lovely and white and smooth when they start off, but once GVHD occurs, you can get this awful kind of scruffy thing going on, and it's basically the human immune system attacking the mouse. So this is a paper from Greenblatt a number of years ago, just showing the incidence of this in these mice. So pink here is nice healthy mice, and all the other colors are sick and or dead. And you can see that for the first 12 weeks, everything's fine, and that's just simply because the human immune system takes about 12 weeks to reconstitute into the mouse after the humanization. But at the star here is kind of when you get full reconstitution, then you can see that the incidence of immune disease and death gets quite severe over time. So this can be very limiting. If you have a study that you can do about in 10 or 12 weeks there, you have lots of healthy mice, and the model is fantastic for that short time point. But if you want to go later than that, you start to have a lot of trouble. In contrast, our mice, this is one of our mice at 35 weeks post-transplant, so way out here, where most of these mice would be dead. And you can see this mouse looks just lovely. It's a little bit gray because she's getting old. And this isn't an isolated mouse, so it would be the entire cohort would be looking really good like this. So I can't say that we have zero GVHD. It does differ between different human donors, but for the most part, our GVHD, if we get it at all, starts much later. And the other thing that we don't get at all is that sometimes this GVHD manifests as a type of wasting disease where the mice just kind of shrivel up and die one day, and we don't see any of that at all in our TKO mice. And we really don't know why we don't get it, but we think it has something to do with one of the knockouts in the background strain. Come on. There we go. So just to show you a little bit more the advantage of that longevity. So this was some work that we did with Andrew Badley at the Mayo Clinic, just showing that we can induce latency using ART in our mouse model. Oh dear, I'm not doing well here at all. Do we have IT somewhere? Oh, we're good again. Oh, we've gone too far. All right. We did ART-induced latency. Oh, thank you. That one's just not working very well. So we actually can treat the mice putting ART drugs into the food. So you can see in our little plot here, 4 weeks post-infection, we had lots of mice with lots of virus in their plasma, and then we started to treat them with ART food, and we could do that over 18 weeks. So that's a very long period of time. And we have the vast majority of the mice become inhibited and undetectable. They're viral load at that time point. There's a few that stay up. Some of these are blips, which is something you would see clinically as well. And some are just mice that were very high and were never suppressed to begin with. Very similar to people, we do not cure the infection with ART, which is an important part of the model. But you do see a great suppression of the proviral load that's left in the mouse. Sometimes you can't detect it when the mice are fully suppressed. But as soon as you take those drugs away, we get complete viral recrudescence with all of the mice, and we'll see that proviral load jump up as well. So very nicely models that type of latency in the clinic. And does it over a very long period of time, which is important for cure studies, because some cure studies can take a long time to establish your infection, get that suppression of your viral load, do your intervention, and then finally take that intervention away and see if you have a difference in viral recrudescence. So the longevity model is really good for those types of studies. So the type of therapeutic that we're looking into as a potential right now is interferon alpha. And I'm sure most of you have heard of interferon alpha. It's a classic antiviral cytokine, has effect against HIV. What you may not realize is that humans have 12 different subtypes of interferon alpha. We tend to talk about it as if it's a single entity, but we're typically just talking about interferon alpha 2, which is the one that is approved for the clinic. But what we've been able to show, and others have confirmed since then, is that really the alpha-14 subtype is much better at suppressing not only plasma viral loads, so these are mice that got alpha-14 versus alpha-2. You can see a greater suppression of viral load in the plasma. And they both can suppress proviral load, which kind of makes them of interest for a cure. But again, alpha-14 outperforms alpha-2 for that. So this makes us really interested in alpha-14. Not only how is it doing this, but how can we harness this for a therapeutic in the future? So my PhD student who just talked in the last session also talked last year and told you that we had some really nice results with him looking at interferon alpha-14's impact on T cells. And the way he looked at this was he would infect our TKO-BLT mice with HIV, let that infection progress for 5 weeks. Then we would treat with our interferons for 3 weeks. And then we would look both immediately post-infection and also for multiple time points after the treatment was over. And what we were able to see that the hyperactivation and dysfunction that is very typical in HIV infection in our T cells was well suppressed in our alpha-14 treated animals. So looking a lot more like uninfected, so markers of activation here, both on CD4s and CD8s. We had a much lower frequency of activated T cells. And when we looked at markers of exhaustion and dysfunction on CD8 cells, so multiple different markers that you may recognize, in every single case we found that the mice that had received alpha-14, these markers stayed suppressed to healthy levels, whereas HIV, they were up, and alpha-2 didn't seem to make it worse, but it didn't seem to help either. So very recently, we've also found that alpha-14 seems to be impacting NK cells. And so this is unpublished data. This was just a little kind of side data that came out of another study that we were doing. And we found, interestingly, that the mice that had received alpha-14, all of them had markers of greater activation of their NK cells. So this was both immediately post-infection with a 10-day treatment or a 10-day treatment after an established infection. And here we were able actually to compare with alpha-2. And we saw in multiple cases that alpha-14 seemed to be having this better effect on NK activity. So we're very interested in this and we want to study it further. But the issue is that humanized mice, NK cells are not very well developed. So we do have them in our mice, but they don't look anything like a human. And the reason for this is because we don't have any cross-reactivity between a cytokine kind IL-15, which is incredibly important for the development of NK cells. So that doesn't cross-react between human and mouse. So if you look at flow plots of NK cells in a healthy human, the marker for NK cells is CD56. And we typically see a bright population, and we also see a 56 dim CD16 population, which is kind of the workhorses of the NK cells. They do all this cytolytic activity. If you look at our BLT model, you can see that we don't have any brights at all, and we get this weird kind of 50-50 mix of plus minus CD16 NK cells. So this isn't a new problem. It has been addressed in some of those NSG models, just simply by adding human IL-15 as a transgenic gene into those mouse strains. So we could do that too. We could just use these mice, but we really, really want to retain our TKO advantages. So we've taken a different tact with this. What we've done is we were kind of lazy and we don't want to develop a transgenic strain, and anybody who keeps mouse strains knows it's a lot to deal with if you have multiple strains to manage in your colony. So we didn't want to develop a transgenic strain, and we don't really want to manage one. So instead what we did is we transduced our mice using AAV with human IL-15. And so we can just inject this, and cells all through the mouse become transduced and start producing IL-15. And suddenly with that in our mouse model, we get much better production of NK cells. So looking in liver, spleen, and blood here, you can see these plots are now starting to look a lot more like that human plot that I showed you in the previous slide. So just to quantify that for you a little bit better, we looked over multiple weeks post-humanization, so all the way out to 29 weeks post-humanization. So our original model are these turquoise guys. You can see there's not very good NK cells there, human here in black. And so I can't read the times. I think it's 18 and 26 or something like that. We have really good, similar to human levels of NK cells. And it's around 29 weeks that they maybe start to fall off, but it's still much improved compared to the original model. And then if we look at those subsets as well, you can now see we have a proper bright population that again is comparable to human. And this very important CD16 positive subset now is also reaching human levels. And again, very important because these mediate cytotoxic activities that could be really key to our development of therapies for HIV. So looking at functionality then, we were kind of pleased to say that even though we kind of had a lousy reconstitution of NK cells in our original model, they actually were pretty functional. So just using a cancer cell line to see if these NK cells would kill them. Even our original model as well as our new model, all were able to kill cancer cells quite equivalently. There just weren't a lot of cells there to do it. Where we saw the really big difference is when we start to look at ADCC activity. So this is where antibodies specific, say, to HIV would bind that CD16 receptor on the NK cells and mediate killing. And so you can see the really great difference now because we finally have those NK cells expressing CD16. We are now getting ADCC activity that is comparable to human as well. So we're very excited to go forward this model and start looking at it for our HIV studies. So in summary then, our TKO-BLT model supports a highly reconstituted and functional human immune system that also includes complement. And it's really a good model for long-term HIV infection and ART-induced latency if you're worried about getting GVHD in your humanized mouse model. With our work with alpha-14, we've been able to show that it very clearly is more potent against HIV infection than alpha-2. And not only that, it improves T-cell hyperactivation and exhaustion after treatment in HIV. And you don't need to maybe maintain that treatment all the time. You can take it away, and that is a sustained effect for multiple weeks. We've also found that alpha-14 increases NK cell functionality during HIV infection. And it really looks like our new TKO-BLT model will retain all of the advantages of our original model, but will now have enhanced NK cell development and functionality that we can do our future studies of alpha-14 mediated effects in HIV. And with that, I'd like to thank everyone involved. So I have underlined here the two individuals with the data that I showed. So Shatu here is making the new NK model. Sarav did the previous work with the T cells, and he talked earlier. I have two new students working on other viruses. I have really great collaborators in Germany who produce the huge amounts of interferon that we require to do our studies. People involved in originally producing the model, and of course the Mayo Clinic that helped us set up that latency model. And last but not least, the wonderful funding agencies that make all of it possible. So thank you.
Great. Thanks again, Kerry, for that great talk. Our next speaker is Dr. Thomas Murooka from the University of Manitoba. Thomas?
Thank you very much. Can you hear me okay? Yeah. All right, sounds good. So again, thank you very much for the opportunity to speak here. So in the next 17 minutes or so, I'm going to go through a couple of things with you. No conflict of interest to disclose. So in terms of introduction, obviously everybody here in the room is quite aware that host immunity really responds to and in constant immunosurveillance for microbial pathogens, and it could range from viruses, bacteria, fungi, and parasites. And in most cases, the immune response is able to respond, eliminate, and generate a long-term immunity. But of course, in certain cases, these pathogens can evolve ways to subvert the immune system in order to establish a chronic and persistent infection. And we're particularly interested in this interaction, but I also wanted to emphasize that the environmental context in which these interactions occur plays a big role, and the genetic makeup of the host response also plays a role. So in my lab, we're really interested in trying to model human disease in animal models, and really kind of going from the animal down to the constituents at the molecular and the cellular level to understand the mechanism. And also, it's a sliding scale. You could go from molecular components and then kind of understand how it contributes to disease at the tissue and the organismal level. So just in terms of learning objectives, I'm going to talk about the interplay between the host immunity, vaginal microbiome, and the barrier function within the lower FGT during health and inflammation, and how to take that information and for us to understand about HIV acquisition in vivo. So many years ago, we were particularly interested in the mucosal tissue, mucosal immunology of the lower female genital tract. And as you, would be aware, it's quite complex. There's structural components by the vaginal epithelial layer. We heard earlier about antibodies, antibacterial antibodies that are produced that are mixed in with mucus. And you can see from this figure that there's a vast array of immune cells that are found within this space. And what I wanted to emphasize here is that this is a highly dynamic environment. Cells are coming in, some cells are coming out, right? So, a lot of chemokines and chemokine receptors are controlling this dynamic process. These kinds of images, 2015 often depicts these dendritic cells as having these long necks that kind of pokes out into the vaginal lumen here. And so, a number of years ago, we wanted to test that and see whether vaginal dendritic cells were able to poke through and sample the lumen. So this is intravital microscopy we did with Sharik at Mass Gen Hospital. This is using mice, right? So these are C11CM cherry mice to label vaginal dendritic cells. And this is a phase where the animals are progesterone dominant, right? So it's very thin. And in this situation, the dendritic cells, number one, are highly, dendritic, right? So, and then number two, they are able to start to interact and capture these HIV particles, these green particles that are found that we placed in the lumen. So the main figure in the middle here is within 1 hour of luminal HIV challenge, and you can see on the right here, this is after 5 hours, right? So the viral particles that these dendritic cells are picking up seems to start to coalesce into clusters, and we've subsequently showed that these dendritic cells can capture the HIV particles and still remain motile within a 3D structural environment. And in data that I'm not going to be able to show you, we showed that in the BLT system, that if you actually block these dendritic cells from coming out of the vaginal tissue, then you can really delay viremia in these animals. So we think that this is an important mode of dissemination. But when we do the same experiment, right, but this time we have imaged animals that were in the estrus phase, right? So when the vaginal epithelium is at its thickest, the picture is a little bit different, right? So first of all, there's a black void here. It's not empty, right? This is where the vaginal epithelium is. We just don't see it. You see the dendritic cells that are found either in the epithelial layer or in the submucosa, and then all the viruses are in the lumen, right? So in these studies, we were able to demonstrate that this vaginal epithelial barrier was creating a significant physical barrier that prevents the luminal contents to come into direct contact with the immune cells. Okay? So I think that was a nice kind of segue to really understand and appreciate the role of the vaginal epithelial barrier in the context of HIV infection or transmission. And we and others have contributed to a field where the vaginal microbiome seems to also play a tremendous role in controlling mucosal inflammation within this site. This is just a diagram from the gut. As many of you are aware, you have vaginal or commensal bacteria. Try to go backwards. That are able to interact with enterocytes, and by virtue of their interactions leads to a response by enterocytes and subsequently responses and inflammatory responses by underlying immune cells. And like I mentioned, there's more accumulating data now that vaginal microbiome in their interaction with the vaginal epithelial cells can also modulate the immune response at this site. This is work from Doug Kwan's lab, where they identified that in women with a lactobacillus dominant microbiome, compared to those individuals, those that had a Prevotella dominant microbiome had transcriptional signatures that overlapped significantly with these LPS-treated monocytes and dendritic cells. So this indicated that, again, the composition of the microbiome seems to be controlling the underlying immune response, possibly through APCs. This is work from Adam Bergner and Lyle McKinnon and Laura and many others from the Caprisa 004 cohort that also sampled vaginal secretions and biopsies and looked at community state types in the microbiome. And they also see different groupings, CT1, CT2, CT3, and CT4. CT1 being Lactobacillus crispatus dominant. CT2 is Lactobacillus inners dominant. And CT3 is G. vaginalis dominant, and CT4 is this polymicrobial microbiota that's dominant with things like Modoboloncus, Prevotella, Smithia, and others. And what's really quite interesting was that if you look at the proteomic signatures of epithelial barrier disruption, or EBD, individuals in the CT4 group had higher signatures compared to CT1. And more importantly for us, they saw a significant upregulation of neutrophil-related pathways, specifically in the CT3 and the CT4 group compared to the CT1 group. And the significance of this was that individuals that had high neutrophil-related factors had a higher probability of HIV infection compared to those that had low neutrophil-related factors. Okay? So this indicated that perhaps neutrophils and the vaginal microbiome and the epithelial barrier function individually contributed to increased HIV acquisition risk. There are studies also showing relationships between these. So neutrophils could be modulated by the microbiome, and the epithelial barrier function also by the microbiome. But what we thought there was a gap in knowledge was whether or not these neutrophils and the activation of these neutrophils had a direct role in barrier function. So to do that, we turned to an animal mouse model to try to model the BV-like vaginal inflammation that we see in these cohorts. And to do that, this is led by Marina. She's a PhD student in my lab. First of all, these were bulb C mice. We also did it with black 6 mice. They're first synchronized with estrogen, right, to synchronize all the animals to the same estrus phase. And then they were subsequently challenged with either PBS or Lactobacillus crispatus, so this is the optimal microbiome, and then the anaerobes that were the Modoboloncus and the Gardnerella vaginalis. 2 times, and at 7 days, the animals were sacrificed and the FGT was harvested. So one of the first things we looked at was neutrophilic response, and indeed, in the presence of these anaerobes here, there was significant neutrophilic recruitment in the vaginal tissues, specifically in the epithelial cells, but also in the lamina propria, and we never saw this in the PBS and the Lactobacillus control groups. And the quantitations on the right. What was quite interesting was to also measure vaginal epithelial barrier function, and you could do that by using a dye that you place in the vaginal lumen, and you look at how much the dye passes through. So very similar to the dextran, fluorescent dextran that people use, but here we're using a dye called lucifer yellow. But also you could appreciate here is that in both cases, animals that were challenged with these anaerobes had a significant disruption of the barrier, evidenced by a number of dyes coming through. One of the questions that obviously we had was this neutrophils driving this phenotype, or are they more of a bystander effect? And so we repeat the same experiment with but in this case, we depleted neutrophils using anti-Lysis G antibodies, and this could largely restore the barrier function, right? So this is looking at lucifer yellow leakage, and you can see that in these 2 cases, it's starting to come down. We also looked at cell-to-cell junction molecules. One of them I'm highlighting here is desmoglin. This is a protein that was also downregulated in non-LD individuals in the Capricea cohort. We also see downregulation of this cell-to-cell junction molecule in the animals when they were challenged with 2 anaerobes, and we think that this is also contributing to this barrier dysfunction that we're seeing. And again, if we deplete the neutrophils here, you can actually completely restore the expression of this junction molecule. So that was really cool, and Marina then subsequently looked at the functionality of these neutrophils. And one of the functions of neutrophils is that their ability to form nets, right? This is release of extracellular traps, DNA and proteases that are functions to trap viruses and bacteria at the site of an infection. But we also know that it's highly inflammatory. So we asked whether this was contributing to this pathology. And if you look at net formation using MPO and citrulated histones, you can see here that there's significant amount of net formation that are found in the presence of these two bugs. Again, we don't see them in the PBS and the Lactobacillus crispatus groups. And what Marina then did was then treat these animals with an enzyme called DNase-1. Now, DNase-1 dissolves extracellular DNA and is an effective way of removing nets from tissues. And so she did exactly the same experiment, but this time gave 3 doses of DNase and asked the question, does this reverse any of this dysfunction that we're seeing? And in fact, she does. So in this case here with Mobiloncus muliaris, there's significant leakage from the lumen into the lamina propria. But if she treats it with DNase across all of the tissue, so the ectocervix and the vaginal tissue, she could largely rescue or improve the barrier function, and the quantitation is on the bottom, right? So here, looking at barrier integrity, she could rescue that with DNase treatment, and also using, looking at nets formation. So this indicated that perhaps nets formation was actually directly contributing to this barrier dysfunction that we're seeing. Kind of a side note that she was also interested in this interaction between nets and Th17 cells. There's been a number of studies looking at the role of histones and actually driving a Th17 signature. And as many of you know, activated Th17s are initial targets of HIV infection in the vaginal mucosa. So I thought this was an interesting kind of avenue to look at. And she did look at a number of different cell types, but just to get into the punchline here, she did look at Th17 cells in PAD4 knockout mice. Now these are animals that are missing a PAD4 enzyme where they're not able to netose, right? The neutrophils are not able to netose. And so in these animals, in their challenge with Mobiloncus, there's very little Th17 production in these cells. So this indicated that perhaps the net formation seems to enhance Th17 frequencies within the lower FGT of these mice in the presence of these BV-like bacteria. So just to summarize here, and I threw a lot of data at you, I think what we're trying to do is create a mice model where if you challenge with anaerobes that is associated with BV-like disease, display a number of inflammatory responses that seems to recapitulate key disease features in humans. One is a rapid recruitment of neutrophils and MMP production, damaging of the epithelial barrier, formation of nets, and the interplay with Th17 cells. Okay, so just in the last couple minutes, this is great, but this is actually contribute to increased HIV acquisition risk. So as Kerry already mentioned, we do use a BLT mice. The background of these animals is different in that we use NSG mice, but otherwise they are created similarly with fetal tissue. And what Marina then did is to take these BLT mice and do exactly the same experiments and looked at functionality of the barrier in the presence or absence of Mobiloncus, and we see similar things. So in these model systems, the bacteria was still disrupting this barrier function. And when she looked at the ability of HIV to cross the epithelial barrier using the same HIV GFP reporter particles that we showed earlier, there was a number of viral particles that were able to cross that barrier compared to in animals that were challenged with Mobiloncus compared to L. crispatus. But the real cool thing was that then we could challenge these animals using repeated low-dose transmitted founder virus, Regio. And so again, these BLT mice were synchronized with estrogen. They were either challenged with PBS or Mobiloncus and give repeated weekly challenges, right? So the idea here is to model kind of low levels of repeated challenges in individuals. And the readout here was to look at weekly plasma viremia and the number of challenges required to reach systemic viremia. So this is one example of what we saw. So what was very clear from these studies is that in animals that had were colonized with Mobiloncus muliaris, experienced plasma viremia earlier compared to control animals. Right? And you can see that on the right here. So this really indicated that the inflammatory response and the barrier disruption that we were seeing in the presence of these anaerobes was directly leading to increased HIV transmission events in these animals. So with that, I just wanted to just summarize here in that, animal models of disease can provide mechanistic insights into how women with a non-optimal microbiota are at a higher risk of STIs, and not just HIV, but other STIs, and that's something that we are also looking into. So with that, I'd like to thank the members of my lab, all the collaborators, very important collaborations with Vanessa Poliquin and Alicia, with Adam, with Vlad at the Reagan Institute, all the funding agencies, study participants, animal care staff, and thank you for your attention.
All right, thanks Thomas for that great talk. Our next speaker is Dr. Amy Gilgrass from McMaster University. Amy?
I'd like to thank the organizers for giving me a chance today to talk about my research. No conflicts to report. Now this slide will be very familiar to most of you, but just as a reminder, there's over 39 million people living with HIV, and when you look at the number of new HIV cases per year, this number has remained relatively static over the last 10 years or so. And here you can see there's about, in 2022, 630,000 people died from HIV-related causes. Now, what many of you may not know is that many of these HIV-related deaths were because of TB. TB and HIV have often been called double trouble, and that's because infection with either one in either direction makes both diseases worse. The numbers of HIV and TB co-infection cases are quite high due to the geographic overlap in these two diseases. As you can see here, this is HIV prevalence in new and relapsed TB cases. In particular in South Africa, over 50% of TB cases are actually in HIV-infected individuals. So what is so challenging about HIV-TB co-infection? Basically, there's 1.2 million HIV-positive new TB cases per year, and a person living with HIV actually has 16 to 27 times more risk of developing active TB. This is a risk of primary infection with TB, reactivation of latent TB, or even reinfection after already having latent TB. And even on ART, this risk still remains elevated. So TB is actually the most common presenting illness among people living with HIV and the leading cause of death. And overall, this graph shows that co-infection worsens disease outcomes of both HIV and TB. As you can see here, I can't actually read it, TB infections is on the left, and you can see all TB cases and the numbers that are HIV positive. When you look at deaths, people living with HIV represent a much higher number within this total. So just to remind people maybe about tuberculosis, as it isn't talked about as much at this meeting, this is a bacterium that is spread via aerosols. It's very infectious. You need less than 10 microbes to develop TB. And actually, one quarter to 1/3 of the world is infected with TB. In most cases, this is latent infection and thus isn't spreadable, but all those with latent TB have a 5 to 10% risk per year of developing active TB. As you can see, in 2022, there was actually over 10 million people who developed active TB, 1.3 million people died from it, and it's actually the 13th top cause of death and the second infectious disease killer after COVID. So how can we study complex diseases such as HIV, TB, and HIV-TB co-infection? So of course we've done many seminal clinical studies, but there's limits to what we can do with human studies, and that can include difficulty seeing what's going on in the tissue. We have large animal studies in non-human primates, which have been seminal, but we have to use SIV, and there's issues with ethics as well as with cost for these studies. So ideally, to study pathogens such as this, we would be using small animal models. But unfortunately, as we've heard, normal mouse models are not susceptible to HIV because it's a human trophic pathogen, which leads us to humanized mice. So when someone says a humanized mice, it can mean very different things. You've already heard about two similar but different models. And really the thing about the humanized mouse models we use is that I'd like to say is that each model has its benefits, and you have to find the right model for the question that you're asking. So in my case, we use sources of hematopoietic stem cells. I use cord blood to isolate CD34 stem cells. And then you need an immunocompromised mouse strains. The base models that we've heard a little bit about are NSG or NRG, and these are just different ways of knocking out the mouse immune cells. These are then generally irradiated, and you inject the CD34 cells either into newborn pups in the liver or IV. And then we wait about 3 months for the immune system to reconstitute. Now, these early models, were very useful and still are very useful as they're in their simplicity, as they got T cells, B cells, all of the main immune subsets. Well, not all of them, but we'll talk about some of the limits. But now we're wanting to develop, or we have developed next generation ones. So part of my research has been developing and characterizing new next generation models. And then within these, establishing and characterizing HIV, TB, and HIV-TB co-infection. And also, I've been using these models to look at vaccination, so vaccines for TB with and without the context of HIV. So that's what I'll show you today. So overall, these humanized mouse models are great for looking at pathogenesis, therapeutics, as well as vaccines. So some of the next generation versions of mice that I'm going to mention here are, I have an NRG base strain that's actually transgenic for human HLAs. I think Kerry mentioned this one. So they're transgenic for both HLA class 1 and 2, or they can be. You can also do one or the other, depending on your question. These are usually called, in the rest of the presentation will be called drag A2. Now, the benefit to these are that you have antigen presentation in the context of the immune cells that you put in. So I match my cord blood to the same HLA type as the mouse. So this can lead to better engraftment of cells and increased functionality of immune cells. I also have models such as that we overexpress human IL-15 in, again, to overcome the fact that our base models don't have NK cells. I won't go into that too much because it was well explained. Okay, so here's some initial data on my drag A2 mice. You can see here we have mouse cells by human cells, and these are all the human leukocytes in the blood. We break that down. We can see we have nice levels of T cells, mostly CD4 T cells, but also CD8, lots of B cells, and we have macrophages as well. Now, when we compare the two models, so the first thing was to see how well they do reconstitute compared to the base model of NRGs. So in black, this is NRG mice. In blue, it's the drag A2. And on the left, these are just how many human CD45 or percentage of human CD45 cells are there over time. And you can see that the drag A2s reconstitute to much higher levels than normal NRG mice. Now, because I'm wanting to look at immune responses in these mice, the types of cells there are just as important. And what we can see here is this is 12 weeks post-engraftment. These are just total human leukocytes, which I've already shown are much higher in the drag A2. But you'll also see that the human CD4 T cells, as well as the monocytes, are quite high. And this is perfect for my model in that HIV obviously needs CD4 T cells to infect, and macrophages and monocytes are also important reservoirs, as well as in TB, macrophages in the lung and CD4 T cells are key in control of this pathogen. I'm going to skip over this a little bit in the interest of time, But in my human NSG IL-15 mice, we consistently over time see that instead of the almost 0% in control animals, we get about 10 to 20% reconstitution with NK cells, and these are the numbers over here. So this can be very useful in studies, and we have started preliminary studies looking at HIV infection in these mice, and they're actually pretty hard to infect in comparison to control. Okay, so in terms of disease phenotype, here you can see we've given HIV intravaginally in our mice, and we've followed them over time for about 6 to 8 weeks. We can see in their blood plasma, both the DRAG-A2 and the human NRG have, by 2 weeks post-infection, have fairly high levels that level off and maintain over time. And as well, we see the characteristic drop in CD4 T cells. So these are the NRGs, these are the DRAG, this is pre-infection, and this is after a chronic infection. We see a drop in the CD4 T cells. In most of my studies, I'm very interested in looking at what's going on in the tissues, which is the benefit of models such as these. And you can see here, this is the vaginal tract of these mice. This is the vaginal lumen, the epithelial lining. And underneath in brown, you can see all the CD4 T cells that are lining the mucosa and act as target cells for HIV. After chronic infection, we see a massive reduction in these numbers. Within these models, I've also been able to do interrectal infections as well as IP infections. Similar to Kerry's, I have a model where we use the ART treatment that's currently used in humans in our mice, and we can see an immediate in pink here. As soon as we start administering, we see a big drop in HIV load, and as soon as we stop administering, I don't know if you can see the gray. Yep, okay. As long as you stop administering, we see rebound over time. Lastly, we've looked at all the T cell subsets, and these mice do reconstitute well with the T cell subsets in the tissues, et cetera. And I'm starting a study on IL-17, and one of the first things was to see if IL-17 T cells are depleted in these mice, similar to what we see in humans. And as you can see here, this is IL-17 CD4 T cells, and it's actually, we're looking in the lung, and we do see a massive drop with chronic HIV infection, and this drop is also seen in plasma IL-17. And as I mentioned, I have looked via flow cytometry and also in the lung, as we're interested in this part for HIV-TB co-infection where these pathogens interact. And we see we get all the lung macrophage subsets as well as the T memory and effector subsets. So I've shown that this works for HIV, but we haven't talked about TB. So here we've infected with TB intranasally, and then 4 weeks later we look at the mice. So here's an uninfected lung, looks very nice. This is an MTB infected lung. It's got massive granulomas throughout. And when we look at colony forming units or amount of TB in there, again, with both models, we can see high levels in the lung, as well as dissemination to the spleen, which is a characteristic of TB in humans. In addition, these are actually more representative of human TB than mouse TB in that they form very human-like granulomas. So here we see, this is a granuloma here in H&E. We see the necrotic core. Here, AFB staining is acid-fast bacilli, where we see the actual TB within the granuloma being controlled. And here we actually see in brown again, CD4 T cells. I don't know how good it is for you out there, but there, you'll believe me if you can't see it, there's a CD4 T cell halo around this granuloma trying to control the growth or the growth of the TB in the macrophages. And lastly, I mentioned that we would be looking at co-infection in these mice. In this case here, we've given HIV first, waited 4 weeks for it to establish, and then given TB and followed the mice. you can see here, well, it's easy to see in the lungs. We actually gave a lower dose of TB in this case. So the TB only at this time point did not have massive amounts of granulomas, but in the co-infected group, we did see extensive granulomas. And this was mirrored by the amount of TB growing in these mice. So here we have in the lung, we have TB only versus co-infected, and then both in the spleen and the liver dissemination, and it was higher in co-infected mice, similar to what we see in humans. We quantified the granulomas here and saw the much greater increase in the co-infected. So thus, these are good models, and I will be proceeding to look at how they can be used to look at immune responses in the lung and to try and identify therapeutics, immune therapeutics that would help. The last thing I wanted to talk about is how we've used these mice in looking at vaccines. So with Zozing, we've created an adenoviral vaccine. It's replication defective, and its newness is that it goes against 3 TB antigens, but also we administer it mucosally. The benefit of this type of vaccine is it targets both innate and adaptive immune responses, CD4 and CD8. And in consultation with Dr. Singh, it made us wonder if this could be an ideal vaccine for people living with HIV, in that we don't necessarily rely on CD4 T cells in case there's a reduced reservoir. And these would be the ideal targets, considering that they're so susceptible. So when we look at these mice, this is the first experiment we did. We gave them just saline or immunized and then challenged with TB, no HIV. And you can see that the vaccine, in this case, is tried-chimpad TB. You can see a beautiful pink lung, and in control, we see massive granulomas forming. This is seen again in the histology, and if you can see this AFB, the massive pink is all the TB within this lung. And when we quantified the CFUs, you can easily see a 2-log drop in the TB, which is actually a very protective effect compared to what we've seen with other therapeutics, and a drop again in the spleen. So as I mentioned, I wanted to see if with having HIV first and the effects that this has on the T cell repertoire would affect our ability to vaccinate in these mice. So in this case, we have more groups. So in the first part, we gave HIV or didn't, and then we gave the vaccine 4 weeks later, and after that, challenged with TB. Okay, so we have our TB only group, TB only plus vaccine, HIV TB, and HIV plus vaccine plus TB. A little complicated, sorry. It is all color-coded, so hopefully that helps. What you can see in this graph here is that we have the non-immunized group on the left and the immunized group on the right. Here we have TB only, and here it's HIV plus TB. So you can see that granulomas were forming extensively in the non-immunized group, and in the immunized group, both with and without HIV, we see much less granuloma formation. And when we did the CFUs for this, looked at the levels of TB in the lung, we did see in the non-immunized group, which are the first two, higher levels than we saw in the last two, the HIV or the TB alone group. Thus, it does appear like this could work even with a depleted CD4 reservoir, so we are repeating this to increase numbers. So overall, we can say that these next generation humanized mice can improve on prior models in terms of immune reconstitution and can recapitulate many features of HIV, TB, and HIV-TB co-infection. We can use them for immunized and challenge studies and look at vaccine efficacy. And overall, these can act as a good preclinical bridge to facilitate translation of basic research into the clinic. I'd like to acknowledge all my lab members who participated in all the work, as well as all my funding agencies. Thank you.
All right, thanks, Amy, for that great talk. Our next speaker is Dr. Nicolas Chaumont from the CAHR. Nicolas?
Thank you, Jimmy, and thank you to the organizer for inviting me to give this talk today, and thank you to all of you for staying around. I know it's late, it's sunny outside, and you're probably angry by now. So these are my conflicts, and today, of course, I'm going to talk about the reservoir. So, you know, we don't have a cure for HIV. Antiretroviral therapy does not cure HIV, and we know that because when people stop treatment, the virus rebounds, and the big question is, where are these viral reservoirs? So where are these reservoirs from which the virus can reignite infection? And if I ask you this question, your answer would be in tissues and everywhere, and both answers would be correct. And actually, several studies have shown in the past that HIV can persist in many deep tissues. This is a picture from a study by Sarah Janella at UCSD that was performed on bodies of people who donated their bodies after their death. And we had a similar study starting in Canada a few years ago, which we call the end-of-life study. So far, we have studied 2 participants, and these are the ones I'm going to show you today. This is usually my quiz when I give this talk in the US, and I ask the American people, where is Edmonton and where is Ottawa? And you can imagine that they don't know, but I guess you do know, so it's not that funny here. But anyway, so we had 2 participants again, very generous donators who decided to give their body for end-of-life research. Their story is quite similar. They were both in their 60s. The first one required medical-assisted death, and the second one suffered from B-cell lymphoma. They were both diagnosed during their 80s. And they were both, and that's important, virally suppressed on antiretroviral therapy at the time of death. So I'd like to thank all the CAHR investigators for this study, which would not have been possible, obviously, without the 2 participants, and also with the tremendous help from Jonathan Angel, from Chris Power, and from all CAHR investigators. I cannot name all of them, but Petronella Ancuta, Ligena Bion, Cecilia Kosteniuk, and all these people have really contributed to this work. So we collected different pieces of tissues. I know this is a session on models of HIV infection, and I'm showing you human data, but I think there are things we can do in humans, and I think this is a proof that we can actually use this very particular context of autopsies to answer very basic questions on HIV persistence. So as you can see, we collected quite a large number of tissues from the brain to the testes. I don't know why I picked these 2 examples, but anyway, lungs, liver, and everything. And the first thing we did, the first thing that Julia Ruiz, Caroline Dufour, and Amélie Pagliuzza in my lab performed was a simple PCR for HIV DNA. So the question is, where can you find HIV genomes in the body of people who have been on suppressive ART for many years? And as you can see here, so the different colors are different organs, and the different dots are different pieces of tissues. As you know, for instance, a liver is a big organ, so we decided to pick different pieces from different parts of the organ and make basically an average of the measures. And you can see that these measures actually are quite consistent within a given organ, and total HIV DNA basically was detected in all collected tissues, and the highest frequencies were found in the lymph nodes, the liver, the spleen, and the lungs. The problem with HIV DNA PCR is that it measures a lot of bad things, a lot of dead viruses, because these genomes present gross defects such as deletions and others, and they are therefore not replication competent. So if you only measure HIV DNA, you're measuring a lot of crap. So you want to do something better, and what we did is to perform full genome sequencing. So this is a work that Freddy Caroline developed in the lab. She developed this assay to sequence near full genomes from this autopsy sample. This is a simple nested PCR. Well, simple, not that simple, because we had to work a lot on the optimization of the assay, but when it works, usually it works. And then we use PacBio sequencing to generate full genome sequences. And so, as expected, most of the proviruses we detected in all the different organs were defective with large deletions, and we sequenced 300 genomes from the first participant from Ottawa. And if you classify these genomes according to their integrity or defect, you can focus on the small green part here, which are the intact genomes. On less than 2% of the genomes that were amplified were genetically intact. So that means that when we measure HIV DNA by PCR, we measure 98% of defective genomes. And the intact genomes, the important ones, the one that can cause rebound, they were primarily found in the spleen, as well in the lymph node, and particularly the mediastinal and mesenteric lymph nodes. In the second participant from Edmonton, very different story. this participant had a lot of intact proviruses. And there is probably something very weird here. We don't know if this is because of the B-cell lymphoma or something else, but I would say it's not a typical patient. Here again, we found intact proviruses in obviously the inguinal lymph nodes and duodenum, but also in other tissues. Okay, so these were human data. We are limited when we do human studies. Obviously, we cannot do interventions. It's difficult to study viral rebound, so we decided to take advantage of a non-human primate study that was performed by David Evans and César Trifonet, a postdoc in the lab, actually collected samples from this study. We used the placebo animals in this particular study. We had 5 animals, so very small study, hard to conclude anything on this, but I'll show you the data because I think it's still interesting. We are all primates, but we are not all primatologists, so we did the best we can do on this. There are people in this room who have way better experience than I do on primates, such as Jérôme, who is in the room, but anyway, I'd like to show you this. It's not published data yet. So what happened to these 5 monkeys? They were treated with antiretroviral therapy for a certain period of time, for 29 weeks actually, and we collected extensively tissues just before antiretroviral therapy interruption. And David Evans sequenced the plasma rebound viruses here, and so what we tried to do is to find whether we could find a match of these rebound plasma viruses. We sequenced those viruses with the tissues that were collected just before treatment interruption in an attempt to identify the source of rebound. So the first thing we observed is that the proportion of intact proviruses in these animals is actually much larger than what you would see in humans. A third of these genomes were genetically intact, which we never see in humans. The gut contained a lower proportion of intact genomes compared to lymph nodes and blood. This is a pie chart. In green are the intact genomes, and you can appreciate the difference. So, of course, the big question is, can we find a match between the rebounding viruses, which are in yellow on these trees, and the intact proviruses that we could sequence from the different tissues? And we did find some matches suggesting that we could identify the source of rebound in some of these animals. And interestingly enough, I'm not going to go through all the details, but we found that in 3 out of 5 animals, the sequences of these variants were identical to genetically intact proviral genomes from a lymph node collected pre-ATI, suggesting again that the lymphoid tissue, and particularly the mediastinal and inguinal lymph node, might be a preferential source of our rebound in this model of HIV infection. Okay, another thing we are looking, because tissues are made of cells, is to try to identify what are the cells in which the virus persists, not only at the tissue level, but really at the cellular level. So far, as you know, we don't have a very specific cellular marker that would allow us to identify the rare cells carrying intact and replication-competent proviruses from which HIV rebound during antiretroviral therapy interruption. And we're not even sure if such a marker exists. This is a very complicated slide that just summarized the work that has been done in the past 15 years on trying to identify reservoir markers. And most of these previous studies used HIV DNA as a readout, which largely, as I showed you before, overestimates the size of the reservoir. So there are novel approaches to phenotype reservoir cells that have been published last year by different groups, one from Shu Yu and Matthias Lichtenfeld, and the other one from Mike Betts. But we wanted a different assay that would measure not only the integrity of the proviruses, but also their ability to actually be induced and to produce viral particles. And so to do so, this is the approach we used. I'm going to go briefly through this, but we basically collected blood from people on antiretroviral therapy, and you can stop me now and say, well, Nicolas, you just told us that the reservoir is in tissues, and that you're looking at blood. What are you doing? So the point is that we want to apply this assay to tissues afterwards, but for now, we used it exclusively in blood samples. So we stimulate those cells to reactivate the reservoir, and we stain for P24 using 2 different antibodies to increase the specificity of the staining. And by doing so, we can actually identify a very clean infected cells from the reservoir that have been reactivated, and we can sort these cells in single well in a 96 well plate. So 1 infected cell that has reactivated HIV per well. And we have adapted the near full genome sequencing assay to actually amplify the full genome from this single cells, and then we can classify them. And because this is a flow cytometry-based approach, you can actually record the phenotype of these cells at the same time. So at the end of the day, you can answer the question, what is the phenotype of the cells in which I can find intact genomes? That was the point of this experiment. So Caroline did a lot of work on this, and in most of the markers we looked at, there was actually no specific enrichment. This is a complicated slide, but basically we classify the genomes according to their defects. So you want to focus on the intact ones in green and see whether or not the level of expression of a given marker, which is written at the top of each panel, is different between the green and the defective genomes. And if you look carefully at the data, you'll see that there are only two that were significantly different, which are alpha-4 and beta-1, which are two chains of the integrin alpha-4 beta-1, also called as VLA4. So we were quite excited with this, but of course you always need to validate a finding, and so this is what we did. We sorted CD4 T cells expressing VLA4 or alpha-4 beta-1, and we used a completely different assay, which is still called the reference or the gold standard assay in reservoir studies, the viral outgrowth assay, in which you basically culture the virus. And so we did the multiple deletions and everything, and we found a 27-fold enrichment in replication-competent virus in the VLA4 expressing cells as compared to the VLA4 negative cells. So we think that this marker significantly enriches in HIV, in replication-competent HIV. Having said that, around 30% of your cells express VLA4, so it's not a specific marker. It means that you enrich in VLA4 expressing cells, but not all your VLA4 expressing cells are infected, obviously. So is this a new therapeutic avenue to target reservoirs? Well, I'd like to think so. The good news is that there is an antibody that targets VLA4. It's called natalizumab, and I was quite excited when Caroline came back to the lab and said, well, it's VLA4, the marker of the reservoir. Let's try to block it or do something with it. And we spoke with our colleagues who are neurologists on the same floor, and they told us, well, don't give that to people with HIV because it causes PML, or progressive multifocal leukoencephalopathy. So you don't want to do that. So we said, crap, we need to find something else. Actually, there are new drugs that can target VLA4 that apparently have a better safety profile, so that we would like to explore in the near future. Actually, this has been done in a non-human primate model by Ken Williams, and he gave this anti-alpha-4 antibody to non-human primates infected with HIV, and he was able to show that natalizumab treatment early can stop productive infection of the brain and the gut, and that it prevents the establishment of the tissue reservoir. So I think that's quite a promising strategy, but again, the toxicity profile of natalizumab in humans makes it just an impossible option to move into the clinics right now. I'd like to finish with one slide from the work of Hélène Roux, who is a postdoc in the lab, and she's presenting a poster just to tell you that these HIV-infected cells, or these reservoir cells more exactly, they are very, very bizarre, very weird. They have a very weird phenotype. So at the top of this slide, I'm showing you the phenotype of all CD4 T cells, and these are data from around 10 different people living with HIV. So if I take your CD4 T cells and we stand them and we look at the expression level of granzymes or senescence markers or immune checkpoint blockers or lineage transcription factors, we'll see that overall only minor fractions of your cells will express multiple of these markers. But when you look at infected cells, P24 positive cells, so those cells that have been infected probably quite a long time ago because these people have been on ART for a long period of time, they start to express many different things. So they express not only more granzyme, which is something that has been observed by Yachi, Ho, and others, but also much more senescence markers. So it seems that the reservoir might become senescent with time on antiretroviral therapy, which is not very surprising because these are all cells that stick around, so you expect them to get tired at the end and become senescent. More immune checkpoint blockers, this is something that we had observed in a previous study, and also more lineage transcription factors and quite often multiple expression. So we found these very weird cells that express both BCL6, FOXP3, and TBET. For an immunologist, it's like a nightmare. You never see that usually. So we're not exactly sure what they are, but you can go to Hélène's poster and ask her. So conclusions, HIV persists in many tissues, with the lymphoid tissues being enriched in intact genomes. In non-human primate models, lymph nodes are a common source of rebound. Again, this is a very small study, so we have to be cautious with this. And this is only one model with sheep and monkeys that have not been treated for a very long period of time. CD4 positive T cells expressing VLA4 are highly enriched in inducible and replication competent proviruses. And finally, the reservoir is enriched in senescent CD4 T cells, suggesting that these senolytic drugs, which have been really à la mode during the past 10 years, they may be used to reduce the size of the reservoir. Most importantly, I'd like to acknowledge all the people who have contributed to this work. So in the lab, mostly Caroline Dufour, Rémi Fromentin, Amélie Pagliuzza, César Trifonet, and Hélène Roux. All my colleagues at the CHUM, that includes, of course, Petronella Ancuta, Andrés Finzi, Bertrand. For the autopsy study, Eric, Ali, and of course, Jonathan and Chris. Of course, Jean-Pierre Routy at McGill, who has been instrumental in all these tissue studies since the very beginning, and also our collaborators at Wisconsin, David Evans. And of course, I'd like to thank the funders, CAHR, but most importantly, all the study participants who contributed to this work. Without them, we could not do anything. Thank you.
All right, thank you to our speakers for these wonderful talks and keeping time. We actually have time for questions. So we're going to, the speakers here have a mic, so they're ready for fielding individual questions, or if you have a question that you want to pose to sort of the group that kind of crosses the talks. Jimmy is excited to get his steps in. He's going to be the mic runner. So try to go from left to right side when you ask questions. Or I can also field from up here. So are there any questions? Yeah.
Yeah, a couple of questions actually. First one's for Nicolas. That end-of-life study is fascinating. The question is about, so when people sample the reservoir in blood, it's pretty clear that clonal expansion of, basically one cell with an intact provirus is a big part of how the reservoir is maintained. I wondered in those tissues what evidence you could get for clonal structure, whether there was compartmentalization, are there individual clones of cells in multiple tissues, in one tissue? I know for one of the participants, there's very few intact proviruses, but for the other one, there's quite a lot. Mm-hmm. So that's one question. And then the other question for the people who are developing the mouse models, it's basically the same question. What is the clonal structure of the cells that persist on therapy? And what's the dynamics, the volume of replication in the mouse model? I have the impression that there's a much higher number of proportionately of infected cells in those mouse models, and how closely do they mimic the humans in that regard?
Should I go to the? No, I have my mic. I forgot it. Very good question, Paul. So yes, we see clonal expansions. It's actually much more detailed in the manuscript and in the paper now. So in every single tissue, we see clonal expansions. And more importantly, I think, we see the exact same sequence, proviral sequence, in distant tissues. So there are two different scenarios, either original infection, proliferation, and then migration to different tissues, or migration to a tissue, local proliferation, and remigration to another one. I think within the lymphoid compartment, so that include all the lymph node and the spleen, for instance, it would be quite normal to actually see this second phenomenon of local expansion and remigration. So I don't know exactly what's the process, but yes, there is definitely clonal expansion in every single tissue we've looked at, and often these clonal expansions are shared between distant tissues.
Maybe I could ask the second question. So I haven't seen any studies that looking at clonal expansion in the BLT or the humanized mouse system, but certainly the approach is there, right? So you could infect these animals, you could put them on ART, and then they rebound. So there's a reservoir population, presumably in CD4 T cells that are generated, but I haven't seen any studies looking at clonality in the BLT system to see whether it actually replicates what we see in humans. I would argue that there's probably contributions by cytokines that are driving this expansion, but also the contribution of antigen, right? And so what those antigens are in the mouse system, I'm not sure, but I think that's a really important question to see whether these mouse models could actually replicate that. And then you could ask the question of, can you interrupt, certain mechanisms to prevent that clonal expansion? Answer your question.
I already forgot the other one.
So
What's the other one?
I just had a quick question for Nicola, and then the other three related more to the mouse models. Nicola, with the studies of end of life, how certain are you that they did not stop taking antiretroviral treatment days before, especially with the second patient you presented, because it could very well be, a receding with replication-competent virus?
So that's a very good point, Eric. For the first one, we're actually quite confident because we had a viral load, I believe, and Jonathan is in the room, but like a few days before death. For the second participant, I really thought that he interrupted therapy because seeing so many intact provisos would suggest active viral replication. So Chris might be in the room, I don't know if he is, but anyway, so he actually checked his files, and I don't think we have a viral load like a few days just before death, so we don't have biological data, but we have what the participant told us. And he told us that he continued to take his pills until the end because he knew that he was contributing to this work. So I trust him.
Okay, so sorry, not quickly, but for the other three. Just a question, because I'm always puzzled by BLT mice models in terms of, small lymph node formations and also just the antigen presentation pathway, especially with class 2. And maybe, how effective is that in the BLT mice models?
I mean, in our model, I think we have pretty good presentation, really. The T cell compartment is very, very strong. And it kind of depends on your myeloid compartment. A lot of times ex vivo, we have to provide APCs. It's not like a human where you can take total PBMC and just throw in peptides. So it is a little bit weaker. But overall, I would say the T cell response is quite strong, and both to the infection itself and then for vaccination. Where we really fall apart a little bit is for B cell responses. I mean, it is also better in the BLTs and also the transgenic strains because you have that MHC interaction. But to try to get strong responses from B cells, strong antibody responses is really limiting, but again, could potentially be, people have shown that by replacing some of these cytokine factors, we can augment that as well.
Yeah, I would say that's a limit with, I think, all the current ones is that we can get B cell responses, and I think all three of the models we use have improved B cell responses where we can get antigen switching. But if you look at the amount of antibody in the blood to an infection or a vaccine compared to a human, they're not on the same scale
It's like
whatsoever.
100-fold lower, at least, yeah.
The first part of this, probably something more for Tom. So what is...
Get the mic here.
So for imaging perspectives on, the formation of small lymph nodes, can you identify them effectively in a BLT mouse model for imaging, for example?
Yeah, so the BLT, in that it has the gamma chain knockouts, the formation of lymph node is defective. You can see mesenteric lymph nodes, but in some of the peripheral lymph nodes, it's not there. Now, some of the earlier imaging studies that we did, we actually used a non-SCID background without the gamma chain knockout. And in those animals, they do have lymph nodes. So, there's obviously pros and cons, like people here said, and depending on the question that you're trying to address, there are models available for you, right? As long as you find them. Yeah. And if they're not available, then you have to create new animal models to be able to ask the question. Yeah.
Keith?
Hi. Big voice over my shoulder here. Keith, University of Manitoba. For Nicola, just the VLA4 that you find overexpressed in the reservoirs, what do you think is the mechanism of that? Is it just that they're overexpressed and so they're more likely to remain in tissues as sort of that, sort of the Velcro, the interaction with the VCAMs and other things? It's just because they're overexpressed, they reside in the tissues much longer, or is there more of a sort of active process going on?
So, I think it has to do with the property of VLA4, which, as you know, promotes migration to the brain, the bone marrow, and the inflamed nodes. And I think that's a process by which HIV-infected cells, after 10, 15, 20 years of antiretroviral therapy, can receive survival signals in this immunological niche. So I think the process is really not direct. I think it's, if you express high levels of VLA4, you're probably more likely to survive for a very long period of time, whether you have HIV, whether your cell is infected with HIV or not. So I have absolutely no proof of what I'm saying now. So we are trying to work on this, but I think it's probably providing the cell with a pro-survival signal that would allow it to persist for many, many years.
Give the final question to Jérôme.
No, it's the same way as Eric, because in the mouse model, if we have no B-cell follicle or T-cell and B-cell interaction, in terms of T-cell differentiation and memory cell, it's possible that the reservoir should be a little bit different to what we found in humans in that case. What's your opinion?
I guess that's for you, not for me.
Yeah. So I know they do form all the memory subsets. Now, actually, that's one of the things I'm intending to look at, like what happens when we give HIV, when we give ART and HIV, and what happens to the subsets. So I don't think we have the answer really to that fully.
Yeah. I mean, we did see some of the same sort of permutations, or not permutations, but the hallmarks again. You'll get this exacerbated effector population coming up and things like that, which I think could potentially impact what type of reservoir you have in the end. I think part of the issue is all these mice are immunocompromised. So like Thomas was saying, we don't really have that same antigen exposure that a human would have, and so I think that is potentially would impact the type of reservoir we ultimately have in the mouse model. But I think we could address that. I mean, they can handle certain types of pathogens, without killing them all, to try to drive some of those processes to look a little bit more like human.
All right, please join me in thanking our excellent panel. And a plug for our Basic Sciences plenary, which will be tomorrow morning. Thanks.